How do we develop effective immune therapies to treat cancer? And since I'm a drug development person that works in trying to identify novel agents and bringing them forward, I wanted to go through the clinical trial process that exists. And you've heard about phase 1, phase 2, phase 3 studies, and what those are, and why do we do them the way we do them. And then talk a little bit more. Dr. Shah has already actually said most of the stuff I was going to say, but we'll talk about some of the rationale for immune-based therapies. What are the different types of therapies? And I'll go into the adoptive T cell therapy and the CAR T cells. Also, the results so far, and this I'm going to just talk about some of the results, sort of a general broad overview of immunotherapy, not a comprehensive for every single indication that's been approved. And then what are we doing to improve on where we are right now? What are some of the side effects to these immunotherapies, and what are we focusing on as the next step? So why do we do things the way we do them in the clinical trial process? So in 1906 in the US was the first Pure Food and Drugs Act, which was to protect against misbranding and adulteration of food, drinks, and drugs. In 1938, there was an unfortunate incident where one of the elixirs had a solvent which contained antifreeze, which resulted in death of some of the patients. So the Food, Drug, and Cosmetic Act was enacted, which basically required pre-market proof of safety before anything gets on the shelf. In 1962, and I'm sure you guys remember late 50s, there was the episode with thalidomide being given to women who were pregnant. It resulted in birth defects. So this whole act was relooked at, and an amendment was instituted that basically required sponsors, who are the people who are developing the agent, novel agent, to seek and to demonstrate safety, demonstrate efficacy for whatever they are claiming that the drug does, and that it should be done using adequate and well-controlled clinical trials. And that's where all the phases that you hear about come from when this act was instituted. Also, there was a desire that patients should know what the side effects are of an agent that's on the market. And so the first package insert came about in 1970. And the process was working, but there was a lot of criticism, and has continued on until recently, that it takes too long to develop these drugs. So when we discover something new in the laboratory, we synthesize a new molecule, it takes 12 to 16 years to get to the market. That includes all the preclinical stuff is done, and then the clinical stuff. So one of the things was, once we bring it to clinic and we notice that something is promising, can we do something to expedite the development? And so the FDA responded by these 2 new acts, and there's some excitement about them. But now the FDA can fast track some of these molecules and bring them forward. And as Dr. Shah mentioned, we are actually in an unprecedented phase of oncology research, because a huge number of new agents have been approved for indication of cancer, which even 2 decades ago used to be 1 drug a year and people were happy. Now we have 5, 6 new drugs that are approved per year for different cancers. So this is the clinical trial process that we have settled on, and I have actually modified it to show where we are right now. And I'll tell you a little bit of the differences that have happened in the last probably 5 years. So the phase 1 study is when you're bringing something from the laboratory to the clinic. And so I looked at those trials basically as, when you're giving it to humans, how are you giving it? What dose are you giving it? So you give it to a certain number of patients. You observe them, make sure they're okay, and then you treat the next set of patients at a little bit higher dose, and then observe them. And it's a stepwise process of increasing the dose till you figure out what is the best way to give it to patients. But since we've been doing genomics and we understand a little bit more about the cancer biology, and we have more of these targeted agents, now we have the immunotherapies, we are more and more selecting patients that go into it with the hope that we're able to start seeing activity right in the first trial that we do with the agent. And so phase 1 studies have gone from being like a 12 to 20 patient studies to these a little bit larger studies, but they are trying to address, is this drug really doing what it's supposed to do? and which patient population should we hone down on that would derive the most benefit. Once we see that benefit, then we take it to phase 2, where we try to prove that what we saw in a phase 1 is actually holding true, and there is benefit in that patient population. It used to be there's a lung cancer trial, there's a colon cancer trial, there's a breast cancer trial. Now some trials, depending on the agent, they put them all together based on if they have the quote-unquote target in the cancer. But anytime you get excited about an agent, you need to prove that it's actually better than what's out there already. Are we actually truly making an advance? And the advance may be that it's more effective, or the advance may be that it's less toxic. And so phase 3 studies typically are done where you compare it to an existing standard. And so as I put in some of the patient numbers at the bottom, and that's one of the things we're trying to do is that can we actually in the phase 2 part address some of the questions, including some of the is it better than the standard, and try to go to the FDA sooner than waiting for all those patients to be treated. And then these patients are treated in centers like here, a lot of observation, people are involved in development so they know what to look for in patients. The true test comes when a drug gets on the market and it's out there in the community. So the FDA requires that there are certain safety studies done when they're administered in the community with patients. So this whole process is geared towards first ensuring patient safety, so we do it slowly, and then how can we expedite development of promising agents while maintaining the highest standards of patient safety. So Dr. Shah already went through and mentioned Dr. Kohli's work that led to us thinking about using the immune system to target cancer. And so when we think about, okay, how do we develop these new treatments and how do we harness the immune system, one of the first steps is understanding why doesn't the immune system work? Why doesn't it actually get rid of the cancer? So when you look at the cancer under a microscope, you can see immune cells sitting right next to the cancer cells. And so the first question was, why aren't they being effective? And we do know that cancer is self. It's a cell that's arising from the human body itself. So it's not something like a virus or bacteria that's coming in, which is easier because it's different in appearance, and so the immune system attacks it. But still we know that the cancer cells are different enough, so the immune cells should be reacting to it. So one of the things that we know that the tumors do is these antigens, which are what your immune cells recognize as different and go attack. So the tumor cells will reduce the amount of these antigens on their surface, so the immune cells can't see them. The other is the PD-1 that Dr. Shah mentioned, which is basically certain proteins that even if the immune cell sees it and comes, it tells the immune cell that no, I'm self, don't kill me kind of thing. So that happens. And sometimes tumor cells can produce factors in their surrounding so that all the immune cells that are even there don't work that effectively. So these three mechanisms are what we are trying to focus on when we are trying to develop new therapies, is how can we circumvent all these things that happen. So there are different types of immunotherapies. So I just put a definition for biologic therapies because some people talk about it being biologic therapies. You know, there is a type of treatment that uses substances made from living organisms. It's not something that you synthesize a small molecule to be synthetic. The immunotherapies, there's the adoptive cell transfer treatments, and I'll briefly talk about CAR T cells and these because they've been in the news quite a bit. The immune checkpoint modulators that Dr. Shah referred to, PD-1, anti-PD-1, and anti-CTLA-4, which was the Yervoy. The therapeutic antibodies, what that is this concept of almost like a targeted bomb, which is basically you have the chemotherapy, but one of the problems with the chemotherapy is because of all the side effects, it hits normal cells, you're not able to give enough of a dose that would be more effective than it is. And so what it is you bind it to an antibody, that antibody takes the chemotherapy to the tumor cell and releases it locally at the tumor site. So you reduce the side effects in the rest of the body and increase the concentration of the chemotherapy at the tumor. the hope is making it more effective. There are a lot of clinical trials that are currently evaluating this approach. And we have a couple of drugs on the market that have been successful. The cancer treatment vaccines, one of them is on the market for prostate cancer, and then the interleukin, the IL-2 that Dr. Shah mentioned. So this is the adoptive cell transfer. So I already told you that when you look at the tumor under the microscope, you can see cells there, immune cells with the cancer cells. So one of the first ideas that came is that you take the cells, you take the tumor out, and you take the cells from it, the immune cells, you grow them out, make more of them, and give them back to the patient. The presumption is because the cell is sitting right there with the tumor cell, these are the subsets of cells that probably have the most chance of recognizing the tumor. So let's just strengthen them and give them back. And this is for what's called TIL therapy. This is the first wave of what was done for what's called adoptive cell transfer. The other two things that are done are the basically you take the lymphocytes or the immune cells from the blood and you modify them. So what are CAR T cells? So CAR T cells, there are two actually approvals. One happened yesterday, I think, that the FDA approved. So what are CAR T cells? So these are basically chimeric antigen receptor T cells. And what happens is you take the patient, you put them on a machine for apheresis. So similar to if you go donate platelets. You have to be hooked to a machine for 2-3 hours. It sort of selects out the cell that it needs and gives the rest of the blood back to the patient. So it sort of goes through. And so through this process, they collect a bunch of T cells. Then they take them to the laboratory and say, okay, the tumor has this antigen on it. Can I synthesize the similar counterpart that exists on the T cell to recognize that antigen? So it's a synthetic process where you synthesize what's called a receptor on the surface of T cells, and you genetically modify that T cell. And then you grow it out in the laboratory. So you have a lot of these T cells. And then you give them back to the patient. And that's what's called CAR T cell, and it's adoptive cell transfer because you're taking the cells out and then transferring them back after you modify them. So there are two approvals from the FDA, both against this antigen called CD19. So both different companies, slightly different processes, but they target the same antigen. One of them is approved for leukemia treatment, and one of them for a type of lymphoma. Okay, so when we're thinking about the immune system and developing new treatments, it's not as simple as, okay, there's just this one white flag. There are multiple white flags that exist, and there are multiple things that stimulate the immune system. So the reason why all of us are able to survive through our infancy and adulthood and stuff is because our immune system is so effective at getting rid of the foreign antigens that it sees. And flip side is that it's very good at recognizing self. So once that doesn't happen, there are all these diseases like lupus and rheumatoid arthritis, and those things start happening because the body is not recognizing the self. So it's well known that there is this whole process of that exists, which are molecules that stimulate the immune system and molecules that suppress the immune system. You heard about PD-1 and CTLA-4. So there are drugs approved against both of these targets. Both of them inhibit the immune system. So, and this is the list of approvals, and it's growing for PD-1 and checkpoint inhibitor therapy. And as you can see, it's across multiple tumor types. And I have listed melanoma because part of melanoma is in the adjuvant setting, like I mentioned. So I'm just going to show you some of the trials that have happened and some of the results with it. So nivolumab or Opdivo was tested in non-small cell lung cancer, metastatic non-small cell lung cancer, both squamous and non-squamous, and compared to docetaxel, which is a standard chemotherapy. Higher response rate, responses last longer. So one of the things that we found with immunotherapies is that the patients that respond the response lasts much longer than what we've seen with chemotherapies. So it seems like the immune system is able to prevent the further growth, continues to act even once you stop giving the agent. And this response was seen even in patients that had any level of what's called PD-L1 expression. I'm not going to go into it in detail unless there's a question, but PD-L1 is something on the surface that has been shown to predict for responses. So this is in melanoma. Ipilimumab is Yervoy, the anti-CTLA-4 that had already shown activity in melanoma. And then when they gave a PD-1, it showed better activity, and so it's approved. But one of the things that with the advent of targeted therapies is that we look at the genomics of the tumor. We look at genetic aberrations in the tumor, and we design drugs to target those genetic aberrations. But what was found is that we may not have a drug to target a particular genetic aberration, but if the tumor has a lot of these mutations, it tends to respond better to immune therapies. And so there's been data both in colorectal cancer and now in multiple other tumor types that if they have what we call hypermutated tumors, they would respond to treatment with immunotherapy. This extends not only to what we traditionally call solid tumors, but in the lymphoma realm, in the lymphoproliferative parts, in classical Hodgkin's disease, it's been approved. So how do we improve on what we have? So I mean, I've shown you this is good. There's been responses, but not every patient responds. And so how do we make it better? So this is actually already now FDA approved in the summer of this year, but combination with chemotherapy in first-line treatment of non-small cell lung cancer. So we saw even more response to the checkpoint therapy, and so it's now approved as combination. So carboplatin with pemetrexed with a checkpoint inhibitor. But also, I have shown you this. Now the other question is when we're trying to improve. So we've already shown that okay, now if you hit PD-1, then the immune cells get activated, now can see the tumor, and the eat me signal is there. But then can we actually make the immune system now stronger? Once it can see the cancer cells, can we make it stronger to actually do the job? And so there are multiple trials that are ongoing, looking at the co-stimulatory part of this, where you now strengthen the immune system now that it can see the cancer. And the other, so one of the strategies is we have two pathways that we know already work. One of them is the anti-CTLA-4, the Yervoy or ipilimumab that I showed you, and one is the PD-1. So the combination of that is approved in melanoma. It's more effective than the single agent treatment. But whenever we say more effective, it comes at a price. Because once you're revving up the immune system, you do have increased side effects. And the side effects come from the immune system recognizing the rest of the cells, the other cells, the normal cells. And it can cause inflammation, like Dr. Chow was mentioning, the thyroid problems. It can cause inflammation in multiple organs. And that is where some of the issues come in on who can get the therapy, how long they can get it, and how you quickly recognize these symptoms and manage them so that these treatments remain tolerable. And so, unfortunately, any of these side effects can happen, and that's why the word of caution is, especially for these trials that are not only doing, okay, can we see the tumor cell, but also can we rev up the immune system, that is best done in places that people are aware of the how to manage the side effects, because a lot of this is you can head it off if you recognize it soon enough. Is that it doesn't work in 100% of patients. Why doesn't it work in the fraction of patients that it doesn't work? So even in non-small cell lung cancer, the response rate is 20-30%. What about the rest of them? But also trying to expand it to tumor types where it's not been shown to work as effectively. For instance, in breast cancer, there's a lot of research going on, especially in triple-negative breast cancer, for trying to apply immunotherapy. We do know that breast cancer biology is such that it suppresses the immune system a lot in the local environment. So maybe just this one PD-1 CTLA-4 may not be enough. We may have to do more things to have the immune system be more effective. Also, these combinations with chemotherapy, with other stimulatory molecules. And then there is a lot going on where we're trying to predict and better manage these toxicities. We are also in the process of exploring, just because somebody has an autoimmune disease. So like Dr. Shah mentioned, that's one of the areas of concern, is that somebody has autoimmune disease. You don't want to give them these agents because the chances that the autoimmune disease will flare up. But there's been a lot of concern, mainly from the patient advocacy groups, is like, what if somebody has fairly well-controlled psoriasis or fairly well-controlled dermatomyositis, so one of the autoimmune diseases? Should we penalize them and not give them these immunotherapies, because these immunotherapies are effective against the cancer that they may have developed? And so we are starting a program to study that question.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.